So that is, there is the formation of seed from ovule. The formation of seed from ovule is also a post-fertilization event, so which we will discuss about. So the dicot seed and monocot seed, if you look into the slides, above slide, so dicot seed will be having two cotyledons, and you can find this epicotyle, which are going to form the plumule, and you find the leaves are going to form from that. The hypocotyle embryonal axis, they are further going to develop to form these radicle, the root system. So there is a seed coat. This is a typical structure of a dicotyledonous seed. Monocot seed, what you can notice is, so the monocotyledon, they are also, the fruits are called as caryopsis because the seed coat and fruit wall, they are fused in case of this caryopsis. Maize, we usually take it as an example for this study of monocot seed. So they have this cotyledon, we call it as scutellum, isn't it? And epicotyle, they have this leaf primordia and the stem shoot axis, which is covered by the coleorhiza. So the hypocotyle has these radicle and the uh, root cap, which is covered by the coleorhiza. So this you notice in the monocotyledonous seed, and you can also see the endosperm in case of this maize plant. So what is seed? Seed is the fertilized ovule, and it is formed within the fruit. That is the characteristic feature of angiosperm salva. Angio means enclosed. Sperma means seed. The seeds are enclosed by fruits. So seed, what is the definition of seed? It is a fertilized ovule which is formed inside the fruit. You know that the ovary develops to form the fruit. Ovule is going to develop to form the seed. So the seed is the final product of sexual reproduction. You should understand that seed is the final product of sexual reproduction. So the seed is the fertilized ovule. It is the final product of sexual reproduction. Seed, what are its components? Andre seed coat irate, cotyledons irate, embryo axis irate. So next time, don't be under the impression the seeds are non-living things. They have this embryo, dormant embryo. If you had put them into soil, they would have developed to form new plant. Okay, so many of us are under the impression seeds are non-living things. Okay, but they are living things. They have this embryo. So that you should understand about, whether it is paddy, whether it is wheat or ragi or any seed for that matter, the pulses or the cereals, they're all living things. They have a dormant embryo. So seed is a final product of sexual reproduction. So seed consists of seed coats. What forms the seed coat? The ovule had integuments, isn't it? So integuments are going to form the seed coat. Some of the ovules, they have two integuments. So after fertilization, the outer integuments forms the testa and the inner integument forms the tegmen. So testa and tegmen are the seed coat. So the seed coat is formed from the integument. And you should also remember the seed has cotyledons. Cotyledons has food material, reserve food material. Still the seed develops to form a leaf and they can undergo photosynthesis. They require food that is present in the cotyledons. Cotyledon has that food for the developing embryo, and they also have this embryo axis, epicotyle and hypocotyle. Epicotyle is going to form the future plumule or the shoot system. Hypocotyle is going to form the future, forms the radicle, and it forms the future root system. Okay, cotyledons are simple, generally thick and swollen. So due to the storage of food, so the cotyledons are generally simple, they are thick and swollen due to storage of food, as in case of legumes. The kalu variety or the pulses legumes, they all have this food stored in the cotyledon, and we eat them for their cotyledons. Even when we are eating cashew nut is cotyledons that we are eating as cashew nut or godambi. Okay, so remember about this seed. Seed is the fertilized ovule formed inside the fruit. It is the final product of sexual reproduction. So what are the components of seed? It has seed coat. Seed coat is formed from integument. If they had two integuments in the ovule, the outer integuments forms the testa, inner integument forms the tegmen. Okay, so the cotyledons are simple, they are thick and swollen due to the storage of food, as in case of this pulses or legumes. So the mature seeds are of two types: non-albuminous seeds, or you can also call it as endospermous, non-endospermous seeds, and albuminous seeds, which have endosperm. So I always ask my students to remember this as full meals and uh, the plate meals. See, full meals, whenever you go to a Darshini or hotel and you go for full meals, you can have unlimited food. So the more you want to have, you'll get they would serve you the rice and the food. But in plate meals, it is restricted. So whatever is given in within the plate, only that you have to utilize. So non-albuminous seeds or endospermous seeds, they have limited endosperm, so that will be utilized for the development of embryo. There is no excess endosperm there. So they have no residual endosperm in non-albuminous seed or non-endospermous seeds, as the endosperm is completely consumed during the embryo development. So this is like plate meals. Okay. Example for this non-albuminous seeds are pea. Pea andre batani, groundnut or peanut, kadlekai, beans. You notice they are all having this non-albuminous seeds because they do not have any residual endosperm. So they do not have residual endosperm. But in albuminous seeds, they have some endosperm. Even after the development of embryo, they have some endosperm. Example in cereals like wheat, maize, rice also, rice endosperm. We are eating the endosperm part of it. So the wheat, maize, barley, castor ಅಂದ್ರೆ ಅರಳೆಣ್ಣೆ ತೆಗೆಯುವಂಥದ್ದು ಅರಳಿ ಬೀಜ, then coconut, sunflower, they all have endosperm. So albuminous seeds, they retain endosperm even after the embryo development. So endosperm is retained. So these type of seeds, we call it as albuminous or endospermous seeds. Unlimited meals ಇದ್ದಂಗಿದು. So they retain some endosperm. Example is wheat, maize, barley, castor, coconut and sunflower. ಈ ಎಕ್ಸಾಂಪಲ್ಸ್ ನೆನ್ಪಿಟ್ಕೋಬೇಕು. ಎಕ್ಸಾಮಿನೇಷನ್ಸ್ ಅಲ್ಲಿ ಎಕ್ಸಾಂಪಲ್ ಕೊಟ್ಬಿಟ್ಟು ನಿಮಗೆ ವೆದರ್ ದೇ ಆರ್ ಆಲ್ಬ್ಯುಮಿನಸ್ ಸೀಡ್ಸ್ ಆರ್ ನಾನ್ ಆಲ್ಬ್ಯುಮಿನಸ್ ಸೀಡ್ಸ್ ಅಂತ ಕೇಳ್ತಾರೆ. So most of the cereals and the oil yielding plants like castor, coconut, sunflower, they are all having this endosperm. Okay, so these we have studied about the coconut and the sunflower. They are all endospermous seeds. So these are all only for your understanding. You have this diagram. Paisam sativum andre batani. ಸೊ ಅದರಲ್ಲಿ ಹೇಗೆ ಎಂಡೋಸ್ಪರ್ಮೇ ಇರಲ್ಲ. ಸೊ ದೇ ಆರ್ ಕಂಪ್ಲೀಟ್ಲಿ ಯೂಸ್ ಬೈ ದಿ ಡೆವಲಪಿಂಗ್ ಎಂಬ್ರಿಯೋ. ಮೇಯ್ಸ್ ಅಲ್ಲಿ ನೀವು ಎಂಡೋಸ್ಪರ್ಮ್ ನೋಡ್ಬೋದು. ಮತ್ತೆ ಇಲ್ಲಿ ನೋಡ್ಬೋದು, ಇಲ್ಲಿ ತೆಂಗಿನಕಾಯಲ್ಲಿ ಮೊಳಕೆ ಆಗಿದೆ. Okay, so then the coconut also has this endosperm even after the embryo has developed. Okay, so the sunflower also have seeds where they have endosperm. So the same definitions are here, but different diagrams they have given here. ಕೆಲವು ನಾನು ನಿಮಗೆ ತೋರಿಸ್ಬೇಕಾಯ್ತು. ಎಸ್ಪೆಷಲಿ ಐ ವಾಂಟೆಡ್ ಟು ಶೋ ಯು ದಿ ಕ್ಯಾಸ್ಟರ್ ಸೀಡ್. ಈ ಹರಳೆಣ್ಣೆ ಬೀಜದ್ದು ನೋಡಿ ನೀವು, ಆ ವೈಟ್ ಕಲರ್ ಆಚೆ ಇರುತ್ತಲ್ಲ, ಇದನ್ನ ಕ್ಯಾರಂಕಲ್ ಅಂತ ಕರಿತೀವಿ. The whitish part, we call it as caruncle. You can notice the white part within the seed of castor, it is the endosperm. Okay, so the hypocotyl axis is there. Similarly, you can see the structure of some seeds. Okay, the monocotyledonous seed, you find that they have this scutellum, which is the cotyledon part of it. So they have the plumule protected by coleoptile, and they have this radical protected by the coleorhiza. So the endosperm is noticed. The endosperm and the embryo, they are demarcated by this proteinaceous layer called as alluron layer. In a monocotyledonous seed, you notice the structure, especially maize is the example that they have taken up. Okay, so occasionally in some seeds like black pepper, so mensu, beet, etc., The remnants of nucellus are also persistent. Usually nucellus they degenerate after the formation of seed. So nucellus ಇರಲ್ಲ ಬಟ್ ಕೆಲವು ಇದರಲ್ಲಿ ರೆಮೆನೆಂಟ್ಸ್ ಪರ್ಸಿಸ್ಟ್ ಆಗುತ್ತೆ. ಸೀಡ್ ಆದ್ಮೇಲೂ ನ್ಯೂಸೆಲ್ಲಸ್ ಇರುತ್ತೆ. ಆ ರೆಮೆನೆಂಟ್ಸ್ ಆಫ್ ನ್ಯೂಸೆಲ್ಲಸ್ ವಿಚ್ ಇಸ್ ಪರ್ಸಿಸ್ಟೆಂಟ್ we call it as perisperm. So the persistent remnants of nucellus we call it as perisperm. ಈ ಕ್ವಶನ್ ನಾವು ನೀಟಲ್ಲಿ ಕೇಳಿದ್ವಿ. So the remnants of the nucellus, persistent remnants of nucellus, it is called as perisperm. So remember this, which is noticed in peppers. Pepper seed, you can notice that. So the integuments of ovules, they are going to harden and they form the tough protective seed coat. And this seed coat has a minute opening. We call it as micropyle, through which oxygen and water enters into the seed during germination. So the tough protective coat is seed coat. It is formed by the integuments of ovule after fertilization. So the seed coat has a small pore called as micropyle through which water enters as well as oxygen enters during the germination. Okay, so seeds germinate by the imbibition process. You should be knowing about that also. So seed from ovule, as the seed matures, so you notice that it becomes dry. The seed matures, mature seed, it would be dry. They reduce their water content. So they 10 to 15% moisture is present for their mass. If they have 10 grams of the seed content, of this 10 gram, 10 to 15% of it is moisture or water content. So the seed, when it matures, it becomes dry by reducing water content. So the metabolic activity of the embryo, ಈ ಎಂಬ್ರಿಯೋದು ಮೆಟಬಾಲಿಕ್ ಆಕ್ಟಿವಿಟಿ ಇರುತ್ತಲ್ಲ ಅದು ಸ್ಲೋ ಡೌನ್ ಆಗೋಗುತ್ತೆ. ಸೊ ಅಂಡ್ ದಿ ಸೀಡ್ಸ್ ಎಂಟರ್ ಇನ್ ಟು ಅ ಸ್ಟೇಟ್ ಆಫ್ ಇನ್ಆಕ್ಟಿವಿಟಿ ವಿಚ್ ವಿ ಕಾಲ್ ಇಟ್ ಆಸ್ dormancy. The embryo activity, metabolic activity slows down, and the seeds enter into an inactive state, which we call it as dormancy. This dormancy is seed dormancy. ಕೆಲವು seed dormancy ಒಂದಿನ ಇರ್ಬೋದು, ಕೆಲವು seed dormancy thousands of years ಇರುತ್ತೆ. Like they have seen that date palm seeds which they had obtained 2000 years back when they planted, they have obtained a plant. So the seed dormancy is for long duration of time in certain plants. In lupinous plant, they have obtained a fossil 10,000 years back material of this lupinous seeds which they have obtained when they planted recently. So they have obtained a plant. So it had been dormant for 10,000 years. So the seed, when they mature, it becomes dry and it reduces the water content. So what are the changes happening when the seed becomes mature? You should note that the seeds become dry, they reduce their moisture content to 10 to 15% of their mass weight or the mass. The metabolic activity of the embryo slows down. It enters into a state of inactivity, which we call it as seed dormancy. Under favorable conditions, that is moisture, oxygen, and suitable temperature, the seeds germinate, or else they remain in this inactive state or seed dormancy, you can notice. So the advantages of seeds. What is the advantage of seeds? Uh, why it is more important for angiosperms? The survival value of seeds are much more better since pollination and fertilization are independent of water. See, in case of bryophytes and pteridophytes, they required water for pollination as well as fertilization. That is not required in case of angiosperms, and seed formation is more dependable and it has more survival capability than the water pollinated plants like bryophytes and gymnosperms. So they cannot spread throughout land, whereas the angiosperms they have better survival capability because they have seeds. So since pollination and fertilization in case of angiosperm seeds, they don't require water, seed formation is more dependable. They are more reliable seed formation. And seed formation, it also has better adaptive strategy for dispersal to new habitats. Since they are in dormant stages, their lifespan is more, so they have better adaptive strategies and they can be dispersed to new habitats because seeds are there. And it also helps, the seeds also help the species to colonize in new areas. Okay, because the seeds are there which could be spread by the birds or the insects or man, and the seeds are, you know, that they are in an inactive state, dormant state, seed dormancy is there, so they can be spread into new areas, they can colonize in new areas. That is the advantage of seed. So seeds have food reserves in the form of cotyledons, they have the food reserves. So the young seedlings are nourished, still they are capable of photosynthesis. So the cotyledons will remain with the plant, still the leaves are mature, the leaves are formed and they undergo photosynthesis. So young seedlings are nourished until they are capable of photosynthesis. So these are advantages of seed, having a seed. The hard seed coat protects the young embryo, so which is found within this cotyledons. So when you break the cotyledons, you can find the embryo. So the hard seed coat, they are going to protect the young embryo. So the being products of sexual reproduction, the seeds are product of sexual reproduction, isn't it? So seeds are formed from the ovule. Within the ovule, the zygote is there, the endosperm with the endosperm is formed. They are all formed by double fertilization, and it is sexual reproduction. So they generate new genetic recombination. Genes ವ್ಯತ್ಯಾಸಗಳಿರುತ್ತೆ ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ, ದೇ ಜನರೇಟ್ ನ್ಯೂ ಜೆನೆಟಿಕ್ ರಿಕಾಂಬಿನೇಷನ್ ಅಂಡ್ ವೇರಿಯೇಷನ್ಸ್ ಇತ್ತು ಅಂದ್ರೆ ಎವಲ್ಯೂಷನ್ ಆಗತ್ತೆ. So being products of sexual reproduction, they generate new genetic combination and variation. Seeds are products of sexual reproduction, so they can generate new genetic recombination and variations. So dehydration and dormancy of mature seeds. What happens in the mature seeds? Dehydration and dormancy, the resting period we call it as, of mature seeds are crucial for storage of seeds. Especially the farmers, they select the best seeds and they store it for sowing to the next season. So the storage of seeds is possible because of the dehydration and dormancy of mature seeds. These are crucial for storage of seeds. It can be used as a food throughout the year. See, now we are storing the seeds because they are dry and there is dormancy. We can store the seed for long duration. You have this various pulses stored in your home in bottles, isn't it? It might be the ಹೆಸರು ಕಾಳು, it might be the kadle, nati kadle, or it might be the avrekadu. So you have all alisande kadu. All these you have stored in the bottles, isn't it? As seeds. They don't germinate. So they have this, since the seeds are having this dehydration and dormancy, they can be stored for longer duration. So it can be, we are using that as food throughout the year, the seeds. So it is economically important for us. The seed formation is economically important for human beings because the mature seeds, they would have undergone dehydration and they also have dormancy. So we can store them for longer duration, and throughout the year we can utilize them as a source of food. And we can also sow the seeds for the selected seeds, we can sow them for the next season to raise the crop. So that is the advantage of seed. In a few species of the seeds, the seed lose viability within a few months. Okay, for some of them it might be few days. It varies from species to species. Viability andre their capability to undergo germination. ಈಗ ನೀವು ಒಂದು 40-50 ಬೀಜಗಳು ಹಾಕಿದ್ರೆ ಅದರಲ್ಲಿ ಒಂದು 4-5 ಬೆಳಿಬೋದು. ಬೇರೆ ಕೆಲವು ಇಮ್ಮೆಚೂರ್ ಆಗಿರ್ಬೋದು, ಅಥವಾ ಸಮ್ ಆಫ್ ದೆಮ್ ಮೈಟ್ ಹ್ಯಾವ್ ಬಿಕಮ್ unviable seeds. That is, their expiry date might be over. That is, they are no more viable. Viable andre they cannot undergo germination. So in a few species, the seeds lose their viability within a few months or a few days, some of them. Seeds of many species live for several years. So there are some examples. Some seeds can remain alive for hundreds of years. The oldest is that of a lupine, the Lupinus articus. It was obtained from the Arctic region, the Lupinus articus. So it has remained alive for hundreds of years, 10,000 years it has remained alive. So they have excavated the seed from the Arctic tundra, the oldest lupine, Lupinus arcticus. The seed When they sowed, they germinated and flowered after a storage period of 10,000 years. So they have been viable even after 10,000 years, the seed. So that such examples are also there. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. So 2000-year-old viable seed of date palm, the Phoenix dactylifera carjura. So the 2000-year-old viable seed was obtained. It was excavated during the archaeological excavation at King Herod's palace near the Dead Sea. And when they sowed into the soil the seed of this date palm, so they developed into a new plant. It was 2000 years old, but it was still viable. So this sort of some of the seeds being viable for hundreds of years or thousands of years. So such recorded evidences are also there. So these are all the advantages of seed. So they can be stored for longer duration because they are dehydrated and they have a dormant seeds. They can be used as a food throughout the year. So there are these are all the advantages of the seeds when compared to the other forms. Okay. So the next stage. So seed from ovule we discussed. The next one is fruit from ovary. So the ovary is developed from a fruit. So the ovary, the fertilized ovary forms the fruit. The ripened ovary develops into a fruit. So the fertilized ovary develops into a fruit. So the transformation of ovules into seeds and ovary into fruit ಎರಡು ಜೊತೆಗೆ ನಡೆಯುತ್ತೆ. It proceeds simultaneously. They are taking place together. So the transformation of ovules into seeds, transformation of ovary into fruit wall, both of them are happening together. It is happening simultaneously. Okay, the wall of ovary, the fruit wall or the ovary wall, which we call it as pericarp, the fruit wall, it develops into pericarp. So in mango, you have studied last year fruits. Drupe, you have studied where the pericarp is differentiated into outer leathery epicarp, middle fibrous mesocarp, and inner stony endocarp. In case of drupe, you studied the example of mango and coconut under drupe. So the pericarp is differentiated into different layers. So the wall of the ovary develops into the fruit wall or pericarp, and the fruit may be fleshy. See, in case of orange, cucumber, the apple, then the berries, drupe, they are all considered to be fleshy fruits. So the fruits may be fleshy, as in case of guava, orange, mango, or they can be dry fruits if the fruit wall is dry and membranous, like in case of groundnut, mustard, saswe. The fruit wall is dry and they are not edible. Okay, so the fruits may be fleshy, as in case of guava, orange, mango, and other fruits like lemon, the Hesperidium fruit. They are all okay, and even cucumber. They are all fleshy fruits. Dry fruits, in case of groundnut, mustard, you notice that the fruit wall is dry and they are non-edible. So we notice that in groundnut and mustard. So fruits are of two types. True fruits, where the fruits are developed from ovary, and in this fruit, the fruit develops only from the ovary. The other floral parts degenerate. In true fruits, you notice that the sepals, petals, androecium, they all wither away. Style, stigma, they all wither away in true fruits. They fall off. So the fruits develop from the fertilized ovary. Example, in most of the plants, you find the true fruit. Now, there are examples of false fruit. The very good example for that is apple. In this diagram, you can notice that. So, apple, the membranous part of it, the core of the apple, this is the genuine fruit. So, when they were a flower, the ovary is inferior ovary, and this inferior ovary was covered by this cup-shaped receptacle. When they develop into a fruit, the cup-shaped receptacle or thalamus becomes fleshy. Okay, and the fruit is inside. What we are eating in an apple is the fleshy, juicy receptacle or thalamus, and we are throwing away the true fruit or genuine fruit. So that is what is there in case of apple as well as pear. The wood apple, marcebo antivalla, adu kuda It's a false fruit. In false fruit, the thalamus contributes to the fruit formation, and we think that to be the fruit and consume. But the genuine fruit is this membranous covering of it, which is the core of the apple. So if you take the TS of it, you can see that they are pentacarpellary, pentalocular, and you can also find the seeds. So the false fruits of apple and strawberry. So what we are eating even in strawberry is thalamus. The genuine fruits in strawberry is on the outer side you find small stone-like structures. They are the genuine fruit. Even in pineapple we are eating the inflorescence. Okay, so if so they are all false fruits, and jackfruit also we are eating the perianth. We are not eating the fruit there. So they are fleshy and juicy. So the other floral parts are fleshy and fruity, especially thalamus, which we notice in false fruits. Technically, apple is called as a pome. Pome is a false fruit. P-O-M-E, pome, it's a false fruit. So you can notice the remnants of calyx, the seed within the ovary, that is the core of it, the flower stalk or pedicel. So these are even the strawberry is a false fruit. We are eating the thalamus. The genuine fruit is the membranous covering of it. So it is the genuine fruit it is a little irritating. So when you consume this fleshy thalamus of cashew nut, most of the time you feel like scratching your throat, isn't it? They cause that irritating sensation because of the milky latex. What you find outside the hard wall is the fruit wall. Within that, they have the seed that is the cotyledons, which we use it as godambi or cashew nut. Okay, so this is the thalamus which we are eating even in case of cashew nut. The thalamus is what we are eating it, and it contributes to the fruit formation, whereas the other things they are all dry and we are it is not edible. Okay, so that is about the false fruits that you have to remember. In some species, the fruits are formed without fertilization. Example, banana is a naturally occurring parthenocarpic berry. They might ask you during examination. So give an example for naturally occurring parthenocarpic berry. The example for naturally occurring parthenocarpic berry is banana is an example for naturally occurring parthenocarpic berry. So parthenocarpy can be induced by the application of growth hormones like auxins, gibberellins. If you spray them onto the flowers, you get seedless variety. Now, seed is an hindrance in many of the fruits like in case of watermelon or in grapes. You don't want to have seeds and you want to have a seedless fruit. So the crop can be sprayed with this auxins or gibberellins, which induce parthenocarpy, that is formation of fruits without fertilization. So parthenocarpic fruits are seedless. So they are seedless. So the apomixis and polyembryony will be discussing in the next class. Okay, or I think I'll complete it. We have another 15 minutes. So the next concept after this parthenocarpic fruits are apomixis and polyembryony. Apomixis is formation of fruits without fertilization. So apomixis is production of seeds without fertilization. Formation of fruits without fertilization is parthenocarpy. So formation of seeds without fertilization, we call it as apomixis. It is the production of seeds without fertilization. So you can see this apomixis in the Asteraceae family, sunflower, Savantige, that is chrysanthemum, Tridax. There you can notice this Dendalion. So you can notice this parachute mechanism because of this hairy calyx or pappus in case of this Asteraceae members. So the some of the Asteraceae members and grasses, you notice that there is a formation of seeds without fertilization. So the formation of seeds without fertilization, we call it as apomixis. So it is called as asexual reproduction is mimicry ಮಾಡ್ತಿದೆಯಂತೆ sexual reproduction. So sexual reproduction is being mimicred by the asexual reproduction. Apomixis is a uh, mimicry of sexual reproduction by asexual reproduction process. So apomixis, it is a form of asexual reproduction that mimics sexual reproduction. So how do we develop apomictic seeds?
What
So
is
the
it?
apomixis is, yes. Any doubts you have,
What is
Naveen?
the mix? What is mix? Mix means
I'm
what, sir?
I'm not able to hear you. Disturbance is there.
Mimics. Mimics
ಮಿಮಿಕ್ರಿ
means what,
ಗೊತ್ತಿಲ್ವೇನಪ್ಪ?
sir?
ಇಮಿಟೇಟ್ ಮಾಡೋದು. ಮಿಮಿಕ್ ಅಂದ್ರೆ ಇಮಿಟೇಟ್ ಮಾಡೋದು. ಮಿಮಿಕ್ರಿ ಮಾಡ್ತಾರಲ್ಲ, ಸೊ ಅದೇ ರೀತಿ ಇಲ್ಲಿ ಇಮಿಟೇಟ್ ಮಾಡ್ತಿದ್ಯಂತೆ. ಸೆಕ್ಶುವಲ್ ರೀಪ್ರೊಡಕ್ಷನ್ ನ ಎ ಸೆಕ್ಶುವಲ್ ರೀಪ್ರೊಡಕ್ಷನ್ ಇಸ್ ಇಮಿಟೇಟಿಂಗ್, ನಕಲು ಮಾಡೋದು, ನಕಲು ಮಾಡೋದು, ಕಾಪಿ ಮಾಡುವಂಥದ್ದು. ಓಕೆ, ಎ ಸೆಕ್ಶುವಲ್ ರಿಪ್ರೊಡಕ್ಷನ್ ಇಸ್ ಇಮಿಟೇಟಿಂಗ್ ದಿ ಸೆಕ್ಶುವಲ್ ರಿಪ್ರೊಡಕ್ಷನ್. ಮಿಮಿಕ್ರಿ ನೀವು ಕೇಳಿರ್ತೀರಲ್ವಾ, ಈಗ ಆಕ್ಟರ್ಸ್ ಆಗಿ ಮಿಮಿಕ್ರಿ ಮಾಡ್ತಾರೆ, ಸೊ ಅದನ್ನ ಮಿಮಿಕ್ ಅಂತ ಕರಿತೀವಿ. Okay, so the asexual reproduction is mimicking the sexual reproduction in case of apomixis. So you got development of apomictic seeds. So you got diploid excel in some species. Diploid excel is there. ಹೇಗೆ ಡಿಪ್ಲಾಯ್ಡ್ ಎಕ್ಸೆಲ್ ಆಗುತ್ತೆ ಅಂದ್ರೆ ಮಿಯೋಸಿಸ್ ಆಗದೆ ಇದ್ರೆ ಡಿಪ್ಲಾಯ್ಡ್ ಎಕ್ಸೆಲ್. So diploid excel is formed without reduction division or meiotic cell division, and directly this diploid excel they are going to develop into embryo without fertilization. In some of the species, so the diploid excel is going to be directly developing into the embryo without fertilization. So how is the diploid egg cell formed, Andre? When the megaspore mother cell does not undergo meiosis, so and they are directly forming the diploid egg cell, so we call that as the diploid egg cell which forms without reduction division. They can develop into an embryo. ಈಗ ಆಲ್ರೆಡಿ ಡಿಪ್ಲಾಯ್ಡ್ ಆಗೋಗಿದೆ, ಝೈಗೋಟಲ್ ಡಿಪ್ಲಾಯ್ಡ್ ಆಗೋದು. ಎಕ್ಸೆಲ್ ವುಡ್ ಹ್ಯಾವ್ ಬಿನ್ ಹ್ಯಾಪ್ಲಾಯ್ಡ್, ಸ್ಪರ್ಮ್ ಸೆಲ್ ವುಡ್ ಹ್ಯಾವ್ ಬಿನ್ ಹ್ಯಾಪ್ಲಾಯ್ಡ್, ಫ್ಯೂಸ್ ಆದಾಗ ಡಿಪ್ಲಾಯ್ಡ್ ಝೈಗೋಟ್ ಆಗೋದು. ಬಟ್ ಇಲ್ಲಿ ಎಕ್ಸೆಲ್ ಡಿಪ್ಲಾಯ್ಡ್ ಇದೆ, so there is no need for a formation of zygote or fertilization. So they directly develop into embryo without fertilization in some of the species. Okay, in many species like citrus and mango variety, ಈ ಆರೆಂಜು ಈ ವೆರೈಟೀಸ್ ಮತ್ತೆ ಮ್ಯಾಂಗೋ ವೆರೈಟಿನಲ್ಲಿ ನ್ಯೂಸೆಲ್ಲಸ್ ಇರುತ್ತೆ. ನ್ಯೂಸೆಲ್ಲಸ್ ಇಸ್ ಔಟ್ಸೈಡ್ ದಿ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್. ದಿ ನ್ಯೂಸೆಲ್ಲರ್ ಸೆಲ್ಸ್ ವಿಚ್ ಆರ್ ಸರೌಂಡಿಂಗ್ ದಿ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್, ದೇ ಡಿವೈಡ್ and they are going to protrude into the embryo sac to form embryos. So the nucellus cells are diploid. Okay, this nucellus cells they divide and they protrude into the embryo sac. Embryo sac ಒಳಗೆ protrude ಆಗತ್ತಂತೆ and they form embryos. So ಇಲ್ಲಿ ಡೈರೆಕ್ಟ್ ಆಗಿ ಎಂಬ್ರಿಯೋ ಆಗುತ್ತೆ ವಿತೌಟ್ ಫರ್ಟಿಲೈಸೇಷನ್. ಮೊನ್ನೆ ಯಾರು ಸಿದ್ಲಿಂಗೇಶ್ ಏನೋ ಕೇಳ್ತಿದ್ದಲ್ಲಪ್ಪ, ಪಾಲಿಎಂಬ್ರಿಯಾನಿ. ಸೊ ಅದು ಹೀಗೆ ಆಗೋದು. ಸೊ ನ್ಯೂಸೆಲ್ಲಸ್ ಡಿಪ್ಲಾಯ್ಡ್ ಆಗಿರತ್ತೆ, ಅದೇನಾಗುತ್ತೆ embryo they divide, nucellus divides and then this they enter into the embryo sac, this nucellus diploid cell, and this nucellus diploid cell within the embryo sac they develop to form the embryo without fertilization. Okay, thus each ovule they contain many embryos. ಒಂದೇ ಓವ್ಯೂಲ್ ಅಲ್ಲಿ ತುಂಬಾ ಎಂಬ್ರಿಯೋಗಳಿರುತ್ತೆ, that you can notice here. So occurrence of more than one embryo in a seed is called polyembryony. So apomixis ಅಂದ್ರೆ formation of seeds without fertilization, we call it as apomixis. Parthenocarpy ಅಂದ್ರೆ formation of fruit without fertilization, we call it as parthenocarpy. Apomixis is formation of seeds without fertilization, we call it as apomixis. And apomixis leads to polyembryony. The nucellar cells they multiply, they divide, and they enter into the diploid nucellar cell enters into the embryo sac, and this nucellar cell within the embryo sac they are going to develop to form the embryos without fertilization. Thus, within a single ovule, they have many embryos. Occurrence of more than one embryo in a seed, we call it as polyembryony. So if the seeds from the hybrids, this apomictic seeds from hybrids are sown, ಈಗ ಜನರಲ್ ಆಗಿ ಒಂದು ಹೈಬ್ರಿಡ್ ಪ್ಲಾಂಟ್ ಮಾಡಿರ್ತಾರೆ, ಅದನ್ನ ನೀವು ಬಿತ್ತನೆ ಮಾಡಿದಾಗ ಗಿಡಗಳಾಗತ್ತೆ, ಮತ್ತೆ ನಿಮಗೆ ಆ ಹೈಬ್ರಿಡ್ ಕ್ಯಾರೆಕ್ಟರ್ ಕಳ್ಕೊಂಡ್ಬಿಡ್ತೀರಾ ನೀವು, ಬಿಕಾಸ್ ದೇ ಅಂಡರ್ಗೋ ಅಗೇನ್ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್ ಅಲ್ವಾ. ಸೊ ಯು ಡೋಂಟ್ ಗೆಟ್ ದಿ ಸೇಮ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಆಫ್ ದಟ್ ಸೀಡ್ ದಟ್ ಯು ಹ್ಯಾಡ್ ಪರ್ಚೇಸ್ಡ್. ಹೈಬ್ರಿಡ್ ಸೀಡ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಹೋಗ್ಬಿಡುತ್ತೆ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್ ಆದ್ರೆ. ಸೊ ಇನ್ ಆರ್ಡರ್ ಟು ಅವಾಯ್ಡ್ ದಟ್, ದಿ ಹೈಬ್ರಿಡ್ ಸೀಡ್ ಅಂಡ್ ಮತ್ತೆ ಹೈಬ್ರಿಡ್ ಸೀಡ್ಸ್ ಎಲ್ಲ ಎಕ್ಸ್ಪೆನ್ಸಿವ್. ನೀವು ಪ್ರತಿ ಬಿತ್ತನೆ ಮಾಡ್ಬೇಕಾದ್ರೂ ಪ್ರತಿ ಸೀಸನ್ ಅಲ್ಲೂ ನೀವು ಪರ್ಚೇಸ್ ಮಾಡೋಕ್ಕಾಗಲ್ಲ ರೈತರುಗಳು. ಸೊ that is the reason what they do is, so hybrid seeds are also expensive. So most of the companies hybrids na ಏನ್ ಮಾಡ್ತಾರೆ ಅಂದ್ರೆ hybrid plants na, they make the seeds into apomix so that they can form seeds without fertilization. Okay, as a result the same character of the hybrid is retained, and these seeds they can be sowed in the next season. If the hybrids are made into apomix, there is no segregation of characters in the progeny, hybrid progeny. So there is no cross pollination variations agalla. So farmers can use the same hybrid seeds to raise new crops. So hybrid seeds, if we make them into apomix, so you can utilize it. They are like clones. You can utilize such seeds to obtain the same plants. Okay, we can obtain the hybrid progeny. So farmers can keep on using this hybrid seeds to raise new crops. So this completes our discussion on the sexual reproduction in flowering plants, and all this syllabus will be included. The chapter sexual reproduction in flowering plants completely it would be included in your tomorrow's test. 50 marks irutte biology, 20 marks from physics, 20 marks from chemistry. Okay, on Sunday I'll be including cumulative test. Ella syllabus irutte. So the reproduction in organisms, then the asexual reproduction, sexual reproduction, that also will be having. Then reproduction in flowering plants, sexual reproduction in flowering plants. Complete syllabus would be given in the cumulative test. You should be prepared for that. Okay. ಈಗ ನಿಮ್ಗೆ ಏನಾದ್ರು ಡೌಟ್ಸ್ ಕ್ಲಾರಿಫಿಕೇಶನ್ ಇದ್ದಲ್ಲಿ ಕೇಳಿ. ಸೊ ಐ ವಿಲ್ ಹೆಲ್ಪ್ ಯು ಔಟ್ ವಿತ್ ದೋಸ್ ಕ್ಲಿಯರಿಂಗ್ ದೋಸ್ ಡೌಟ್ಸ್. ಏನಾದ್ರು ಡೌಟ್ಸ್ ಇದೆಯಾ? Do you have any doubts or clarifications?